Welcome to the PubMed Daily Review for May 26, 2026. Today we scanned 291 new open access papers across 137 journals. In this video, we'll cover 10 trending research topics and 10 selected papers for detailed analysis. Trend 1 of 10, Biomaterials, Upcycling, and Drug Delivery. This trend explores biomaterial development, upcycling waste into valuable products, and advanced drug delivery systems. This cluster covers 17 papers. Representative work includes primary challenges in the upcycling of genetically modified spent microbial biomass, phosphate removal, recovery, and upcycling by wollastonite and calcium phosphate mineral hydrogel composites, and multifunctional hybrid lipid polymeric nanoparticle enabling resveratrol and Cernico delivery for enhanced cutaneous wound repair. Trend 2 of 10, Diverse Biomedical and Cognitive Studies. This trend combines research across biomedical mechanisms, clinical conditions, and cognitive processes, reflecting a broad scientific scope. This cluster covers 21 papers. Representative work includes the role of RNA modifications in the pathological mechanisms and therapeutic targeting of multiple myeloma, neonatal acute kidney injury, diagnosis, recognition, and prevention, and the architecture of curiosity, how spatial configuration influences affect, exploration, and memory. Trend 3 of 10, Clinical Trials and Disease Management. Investigating disease management strategies through clinical trials, treatment evaluations, and biomarker research. This cluster covers 15 papers. Representative work includes programmed intermittent epidural bolus versus continuous epidural infusion combined with patient-controlled epidural analgesia for postoperative analgesia, a meta-analysis of randomized controlled trials, ARDS and corticosteroids, beyond COVID-19, and neuromuscular exercise combined with pain neuroscience education for knee osteoarthritis management in older women. Protocol for a randomized controlled trial. Trend 4 of 10, Health, Comorbidities, and Lifestyle Factors. Researching health conditions, comorbidities, and the influence of lifestyle factors on well-being. This cluster covers 28 papers. Representative work includes neurodevelopmental and psychiatric comorbidities of children and adolescents with specific learning disorders, associations with gender and educational level, iron, autoimmunity, and thrombosis, exploring sex-specific interactions, and measuring gambling harm in self-reported questionnaires, a scoping review. Trend 5 of 10, Molecular Oncology and Cancer Targeting. This trend explores molecular mechanisms, diagnostic markers, and therapeutic strategies for various cancers. This cluster covers 48 papers. Representative work includes targeting EPCM expression via near-infrared fluorescent antibodies enables microscopic delineation of primary and recurrent HNSCC. Integrative machine learning reveals interplay between epithelial and endothelial cells elevates mixed heavy metal-related colorectal cancer risk, and sexually dimorphic roles of toll-like receptors in the central nervous system. Trend 6 of 10, Metabolism, Proteomics, and Microbial Dynamics. This trend explores metabolic, proteomic, and microbial changes across diverse biological systems and conditions. This cluster covers 17 papers. Representative work includes blocking beta-alanine synthesis triggers widespread perturbations of energy and lipid metabolism in the brain, proteomic response of Staphylococcus aureus exposed to nisin and liposome-encapsulated nisin, and identification of serum biomarkers in acute aortic dissection using tissue-informed metabolomics methods. Trend 7 of 10, Diverse Biomedical and Physiological Studies. This trend encompasses varied research across animal and human physiology, pathology, and biomedical engineering applications. This cluster covers 34 papers. Representative work includes activation of the RAS/PAR pathway reduces backfat deposition of Gale, Bos frontalis. Preclinical dental students' awareness and integration of pulp biology knowledge. A cross-sectional study assessing the understanding of pulpal functions and implications for clinical endodontic competency and practice, and systemic lipid abnormalities and choriocapillaris flow deficits in patients with hard drusen. Trend 8 of 10, Epidemiology, Health Outcomes, and Modeling. This trend examines health outcomes, disease distribution, and predictive modeling across diverse populations and biological systems. This cluster covers 32 papers. Representative work includes temporal trends and forecasted cardiac arrest and hypertension-related mortality in U.S. adults, 2000 to 2035, a nationwide CDC Wonder multiple cause of death study. predictors of 1-year visual outcome in central involving submacular hemorrhage secondary to neovascular AMD, and prevalence of periodontal diseases in children and adolescents in Asian countries, a scoping review. Trend 9 of 10, AI-driven prediction and optimization. Leveraging AI and machine learning techniques for forecasting outcomes and enhancing system performance across diverse domains. This cluster covers 32 papers. Representative work includes machine learning prediction of dual absorber lead-free perovskite solar cells for boosting PCE, deep learning-based construction of a high-resolution earthquake catalog for the 2023 GISHISHN formula, C-text, 6.2 earthquake sequence, and comparative assessment of radiocarbon and balance method for determining biogenic and fossil CO2 emissions from waste-to-energy plants in Austria and China. Trend 10 of 10, healthcare practices and medical guidelines. This trend explores the development, evaluation, and implementation of medical practices, clinical guidelines, and healthcare professional education. This cluster covers 47 papers. Representative work includes spotlight on mentoring undergraduate medical students using evidence-based medicine, lipid clinics worldwide, harmonization and guidance on how to optimally organize and fund. European Atherosclerosis Society consensus statement across 55 countries and more than 500 lipid clinics, an association analysis of Helicobacter pylori infection with human genetic polymorphisms and serum 25-hydroxyvitamin D3 levels. Up next, our first paper out of 10, Lipid Clinics Worldwide, Harmonization and Guidance on How to Optimally Organize and Fund. European Atherosclerosis Society consensus statement across 55 countries and more than 500 lipid clinics by Christian Bork and other national leads for the EAS Lipid Clinic Network from Department of Cardiology, Aalborg University Hospital, Gistrup, Denmark, Department of Clinical Medicine, Aalborg University, Aalborg, Denmark. This paper was selected because it represents a critical international consensus statement from a highly reputable society, providing essential guidance for harmonizing and optimizing lipid clinics worldwide, which directly impacts global public health efforts to prevent and manage atherosclerotic cardiovascular. Let's break down its key contributions. Globally, atherosclerotic cardiovascular disease, or ASCVD, remains a leading cause of death. Specialized lipid clinics play a crucial role in preventing ASCVD by providing advanced care for high-risk patients. However, there's been a significant gap in standardized guidance for how these vital clinics should be organized and funded worldwide. This paper, a consensus statement from the European Atherosclerosis Society, brought together insights from over 500 lipid clinics across 55 countries. The goal was to develop comprehensive recommendations and guidance. Unlike a traditional study, it didn't compare interventions, but rather synthesized expert opinion to establish best practices for lipid clinic organization and funding. The paper delivered several key outputs. It provided detailed recommendations for enhancing education and training in lipid disorders, which is crucial for improving patient care. It also offered clear guidance on optimal staffing structures for lipid clinics, ensuring they are well-equipped. Furthermore, it included vital recommendations for national and regional policymakers regarding funding, aiming to secure the resources needed for these clinics to thrive. Ultimately, the long-term vision is to significantly reduce the societal burden and costs associated with cardiovascular diseases. This consensus statement is incredibly significant because it provides the first comprehensive, harmonized guidance for lipid clinics worldwide. It addresses a critical unmet need, offering a roadmap for improving care for high-risk patients. While these recommendations are a major step forward, their successful implementation will require dedicated efforts from healthcare systems globally. Moving forward, we need to focus on widespread adoption and continuously evaluate how these guidelines impact ASCVD prevention and patient outcomes. Up next, our second paper out of 10, CT adipose measures identify severe SARS-CoV-2 risk beyond traditional obesity metrics. A C4R cohort study by Nadine Al-Namani and Michaela R. Anderson from University of Pennsylvania Perelman School of Medicine, Spruce Street Gates 9WS, Philadelphia, Pennsylvania, USA. This paper was selected because it introduces a novel and clinically actionable method for refining SARS-CoV-2 risk stratification by utilizing CT adipose measures, significantly advancing beyond traditional obesity metrics to address a pressing global health challenge. Let's break down its key contributions. We know that a higher body mass index, or BMI, is associated with more severe outcomes from SARS-CoV-2 infection. However, BMI is a very general measure and doesn't tell us about where fat is distributed in the body. This study aimed to investigate if specific types of adipose tissue, measured by CT scans, could provide a more precise prediction of severe COVID-19 risk, going beyond what BMI alone can tell us. Researchers utilized data from the Collaborative Cohort of Cohorts for COVID-19 Research, or C4R, which included over 8,400 participants from several major U.S. cohort studies. The key exposures were measurements of different adipose tissue depots, like pericardial and visceral fat, taken from CT scans performed years before the pandemic. The primary outcome they tracked was the time until hospitalization or death due to SARS-CoV-2 infection, with follow-up extending for nearly 2 years from March 2020. The study found that higher pericardial adipose tissue, or PAT, was significantly associated with a 62% increased hazard of hospitalization or death from SARS-CoV-2. Similarly, higher abdominal visceral adipose tissue, or VAT, showed a 41% increased hazard. Importantly, these associations remained strong even after adjusting for traditional risk factors like BMI, diabetes, and cardiovascular disease. However, subcutaneous and hepatic fat did not show the same independent link to severe outcomes. This research highlights that specific fat depots, particularly pericardial and visceral adipose tissue, are much better predictors of severe SARS-CoV-2 outcomes than general BMI. This suggests that relying solely on BMI might miss important risk factors in some patients. Moving forward, these CT-based adipose measures could potentially be used to identify individuals at higher risk for severe disease from various viral infections, and further research is needed to understand the biological mechanisms linking these fat depots to lung injury. Up next, our third paper out of 10, Nuclear KRT19 links the NF-kappa-B-FSCN1 signaling to gastric cancer metastasis by Jiajie Zhou and Daron Wang from Northern Jiangsu People's Hospital, Clinical Teaching Hospital of Medical School, Nanjing University, Yangzhou, China. This paper was selected because it identifies a novel molecular mechanism involving nuclear KRT19 and the NF-kappa-B-FSCN1 signaling pathway that drives gastric cancer metastasis, a critical and difficult challenge in oncology. Let's break down its key contributions. Cancer metastasis is a devastating process, responsible for the majority of cancer-related deaths. Despite its critical impact, our understanding of the molecular drivers, especially the role of structural proteins like keratins, is still evolving. This research aimed to shed light on how keratin 19, or KRT19, contributes to the spread of gastric cancer and the specific pathways it influences. To investigate KRT19's role, researchers employed a multifaceted approach. They utilized both gastric cancer cell lines for in vitro experiments and BLB/C-nude mice for in vivo metastasis models. Key techniques included transcriptomic analyses to identify KRT19's relevance, alongside functional assays measuring cell migration and invasion. They also performed detailed molecular studies to uncover the specific signaling pathways involved in KRT19's effects. The findings were striking. Clinically, KRT19 expression was markedly elevated in gastric cancer patients and correlated with more aggressive disease features. Functionally, depleting KRT19 in cancer cells impaired their ability to migrate, invade, and form spheroids. Crucially, in animal models, silencing KRT19 significantly reduced hepatic metastatic colonization. Mechanistically, nuclear KRT19 was found to sustain NF-kappa-B activity by degrading IB, which in turn enhanced FSCN1 transcription, driving metastasis. This research provides crucial insights into how an intermediate filament protein, KRT19, can rewire cellular programs to drive metastasis. It not only defines a novel signaling cascade involving NF-kappa-B and FSCN1, but also positions epithelial structural proteins as underappreciated drivers of cancer spread. These findings open exciting avenues for developing KRT19 targeted therapies to combat gastric cancer metastasis, urging further investigation into their clinical potential. Up next, our fourth paper out of 10, Dynapenic abdominal obesity and elevated risk of multidimensional multimorbidity across physical, psychological, and cognitive domains. Evidence from longitudinal cohorts by Shiqiang Zhang and Hua Meng from Capital Medical University, China-Japan Friendship Hospital. This paper was selected because it identifies dynapenic abdominal obesity as a significant risk factor for multidimensional multimorbidity across physical, psychological, and cognitive domains, utilizing robust longitudinal data from large human cohorts, which addresses a pressing. Let's break down its key contributions. Today, we're exploring a critical health challenge, multidimensional multimorbidity, or MMM, which involves multiple health issues across different domains. The long-term impact of a specific condition, dienapenic abdominal obesity, or D/AO, on MMM has been unclear. This research aimed to shed light on this important gap, investigating how the combination of muscle weakness and central adiposity affects overall health trajectories. Researchers conducted a longitudinal cohort study, analyzing data from over 8,300 adults aged 45 and older across two major studies, Charles in China and HRS in the US. They focused on participants who were free of multidimensional multimorbidity at the start. The primary exposure was dienapenic abdominal obesity, and they tracked the development and progression of various MMM patterns over a 4-year period. This robust design allowed for cross-cultural comparisons and a deep dive into long-term health trajectories. The findings were striking. Dienapenic abdominal obesity was significantly linked to an increased risk of all 3 MMM patterns. The strongest association was for the most complex pattern, PPCMM, with odds ratios of 2.475 in CHARLS and a remarkable 5.712 in HRS. We also observed that D/AO was associated with a higher likelihood of entering incident/worsening trajectories, with relative risk ratios around 2.0 to 2.5. Interestingly, this risk was markedly higher among individuals under 65 years old, suggesting a critical window for intervention. This study provides crucial evidence that dienapenic abdominal obesity is not just a comorbidity, but a significant longitudinal risk factor for developing and worsening multidimensional multimorbidity, especially in older adults. This suggests a powerful opportunity for early intervention. We should consider integrating simple assessments of muscle strength and abdominal adiposity into routine primary care to identify at-risk individuals sooner. Moving forward, the next step is to conduct interventional studies to confirm if targeting D/AO can effectively reduce the burden of multimorbidity. Up next, our 5th paper out of 10, Association of Long COVID with Major Adverse Cardiovascular Events and Mortality, a real-world data cohort study by Yuting Tsai and Yingqing Chun from Institute of Medicine, Chung-Shan Medical University, Taichung, Taiwan. This paper was selected because it addresses a critical and rapidly evolving global health crisis by providing real-world evidence on the association between long COVID and severe cardiovascular events and mortality, significantly advancing our understanding of its. Let's break down its key contributions. Hello, everyone. Today, we're diving into a critical area of post-COVID health, the long-term effects of long COVID. While we've learned a lot about the acute phase of COVID-19, the lingering health challenges, especially concerning the cardiovascular system and overall survival, remain a significant concern. This study aimed to shed light on whether long COVID truly elevates the risk of serious cardiovascular events and mortality. This was a retrospective cohort study leveraging the vast TriNetX real-world data network. Researchers included adult patients diagnosed with COVID-19 between 2020 and 2023. The exposure group consisted of individuals diagnosed with long COVID within 3 to 6 months after their initial infection, compared to COVID-19 patients who did not develop long COVID. The team then tracked these groups for the occurrence of MACE and all-cause mortality. The findings were quite striking. Patients with long COVID faced a significantly higher risk of MACE, with a hazard ratio of 4.48. This means they were over 4 times more likely to experience these serious cardiovascular events. Specifically, the risk for coronary artery disease was 6.48 times higher, and for stroke, it was 3.46 times higher. Furthermore, all-cause mortality was also significantly elevated in the long COVID group, showing a hazard ratio of 1.53. This study provides compelling real-world evidence that long COVID is not just about lingering symptoms, but is associated with a substantially increased risk of MACE and mortality. Clinicians should be highly vigilant for cardiovascular complications in patients with long COVID. While this retrospective data is powerful, future prospective research is essential to confirm these findings, understand the biological mechanisms at play, and develop targeted interventions to mitigate these serious risks. Up next, our 6th paper out of 10. Macrophage exosomatified MN/SE nanoheterostructure induces post-infarction cardiomyocyte regeneration and immunometabolic microenvironment repair by Linghuan Guo and Yuehua Li from Institute of Diagnostic and Interventional Radiology, Shanghai 6th People's Hospital, Shanghai Jiaotong University School of Medicine, No. 600, Yishan Road, Suwei District, Shanghai, China. This paper was selected because it introduces a highly novel and multidisciplinary nanotherapeutic strategy for cardiomyocyte regeneration and immunometabolic repair after myocardial infarction, addressing a critical and difficult challenge in cardiovascular medicine with significant translational potential. Let's break down its key contributions. Myocardial infarction, or MI, is a leading cause of death globally, and current treatments only offer partial improvements in heart function. There's a critical need for advanced, precise therapies that can truly interrupt the vicious cycle of damage after a heart attack. This research aimed to address that challenge by developing a novel nanotherapeutic system to promote heart repair and regeneration. Researchers designed a novel nanotherapeutic system, PMS, based on a manganese-selenium nanoheterostructure. They modified it with macrophage-derived exosomes to enhance precise targeting to injured myocardial areas. The study investigated how this system impacted cardiomyocyte survival, regeneration, and the overall cardiac microenvironment, specifically looking at the hippo signaling pathway. The results were very promising. The PMS system significantly promoted cardiomyocyte survival, improved extracellular repair, and restored mitochondrial homeostasis. It also effectively suppressed oxidative damage and immune inflammatory disorders. Crucially, the system induced cardiomyocytes to restart their cell cycle, leading to regeneration, primarily by inhibiting the hippo signaling pathway, ultimately preserving cardiac function. This study presents a significant step forward, offering a multifunctional nanotherapeutic system that can induce cardiomyocyte regeneration and repair the cardiac microenvironment after a heart attack. While highly promising, future work will need to focus on validating these results in larger animal models and thoroughly assessing long-term safety before considering human trials. Ultimately, this research provides an exciting blueprint for developing next-generation precision nanotherapies for myocardial infarction. Up next, our 7th paper out of 10, a dual-pathway WNT/IL-13 fusion protein enhances human intestinal regeneration through tough cell activation by Li Yang and Claudia Y. Janda from the Prinses Maxima Center for Pediatric Oncology, Utrecht, the Netherlands. This paper was selected because it addresses a critical unmet medical need in intestinal regeneration for conditions like inflammatory bowel disease and graft-versus-host disease by developing a highly innovative dual-pathway WNTL-. Let's break down its key contributions. Millions of people suffer from severe inflammatory conditions like inflammatory bowel disease, often due to compromised intestinal regeneration. Unfortunately, current treatments are limited, highlighting a critical need for new therapeutic strategies. This research explores whether a novel WNT/IL-13 fusion protein can enhance intestinal regeneration by activating specific cells, potentially offering a new path forward. To address this challenge, researchers engineered a unique WNT/IL-13 fusion protein designed to simultaneously activate both the WNT and IL-4/IL-13 signaling pathways. They then tested this innovative protein using human intestinal organoids, a sophisticated in vitro model. The goal was to see if this dual pathway activation could enhance tough cell activity and promote intestinal regeneration. The study found compelling results. The WNT/IL-13 fusion protein successfully promoted mucosal healing and significantly enhanced intestinal regeneration within the human organoid models. This positive effect was linked to an observed increase in tough cell activity, confirming the protein's intended mechanism. The researchers effectively demonstrated that their dual pathway approach worked as hypothesized. This research presents a significant step forward, demonstrating that a dual pathway WNT/IL-13 fusion protein can effectively promote intestinal regeneration by activating tough cells. While these exciting findings were observed in human organoid models, further in vivo validation is crucial. Ultimately, this innovative strategy holds immense promise for developing much-needed new therapies for debilitating conditions like inflammatory bowel disease and graft-versus-host disease. Up next, our 8th paper out of 10, a heterobifunctional IRAK4 targeting degrader impairs midosome signaling in acute kidney injury and ameliorates kidney fibrosis by Marta Lopez-Marfil and Dario Rubin Lemos from Division of Renal Medicine, Brigham and Women's Hospital, Boston, Massachusetts, USA. This paper was selected as the top choice because it introduces a highly novel and impactful pharmacologic strategy using heterobifunctional degraders to target IRAK4, offering a promising new therapeutic avenue for the. Let's break down its key contributions. Inflammatory signaling, particularly through the midosome complex, plays a critical role in the development of kidney injury and fibrosis. While existing IRAK4 inhibitors have shown some promise in reducing fibrosis, their anti-inflammatory effects are often limited. This study explores a novel approach, using targeted degradation of IRAK4 to more effectively address both inflammation and fibrosis in kidney disease. Researchers investigated a novel heterobifunctional IRAK4 degrader, KTX545, in various models. They used primary kidney fibrogenic cells and human kidney organoids for in vitro studies, and an in vivo model of ischemia/reperfusion injury. The team compared the degrader's effects against a traditional IRAK4 kinase inhibitor, CA4948, to assess its efficacy in reducing inflammation and fibrosis. The results were quite compelling. The IRAK4 degrader, KTX545, successfully abrogated IRAK4 protein levels and significantly reduced myddosome complex formation in kidney cells. In human kidney organoids, we observed a clear reduction in extracellular matrix deposition and fibrogenic gene expression. Most importantly, in an in vivo model, KTX545 showed increased efficacy in ameliorating fibrosis compared to the conventional IRAK4 kinase inhibitor. This study highlights a significant advancement in treating kidney fibrosis. The targeted degradation of IRAK4 offers a novel and potentially more effective therapeutic strategy compared to traditional kinase inhibitors. While these preclinical results are highly promising, the next crucial step will be to conduct further research and clinical trials to bring this innovative approach to patients suffering from renal fibrosing disorders. Up next, our 9th paper out of 10, Microglial CCL4, CCR5 signaling links systemic inflammation to synaptic loss and memory deficits in chronic kidney disease by Yaochen Chao and Ziqiang Wang from Department of Medicine, Hainan Medical University, the first affiliated hospital of Hainan Medical University, number 31 Longhua Road, Haikou, Henan, China. This paper was selected because it identifies a novel and specific molecular pathway, CCL4, CCR5 signaling, linking systemic inflammation from chronic kidney disease to microglial-mediated synaptic loss and cognitive impairment. Let's break down its key contributions. Chronic kidney disease, or CKD, is a growing global health concern, and we're seeing a strong link between CKD and cognitive decline. However, the exact molecular pathways connecting systemic inflammation, a hallmark of CKD, to brain dysfunction and memory problems have remained unclear. This study aimed to shed light on this critical gap, specifically investigating if a particular signaling pathway in brain immune cells plays a role. Researchers used a well-established 5/6 nephrectomy rat model to induce CKD, carefully validating the disease state. They then performed a comprehensive analysis, including various cognitive tests to assess memory, and advanced techniques like single-cell RNA sequencing and immunofluorescence to examine brain changes. Crucially, they manipulated the CCR5 gene using CRISPR technology both in living animals and in cell cultures to understand its functional role in microglial activity. The results were striking. CKD rats exhibited significant cognitive deficits, along with clear evidence of increased microglial activation and synaptic loss in the hippocampus. When the researchers knocked down CCR5, they observed a remarkable reduction in microglial synaptic engulfment, which led to restored synaptic plasticity and, importantly, improved memory function. Conversely, overexpressing CCR5 enhanced this harmful phagocytic activity, confirming its central role. This research provides compelling evidence that the CCL4, CCR5 signaling pathway is a key player in the cognitive decline associated with CKD, directly linking systemic inflammation to synaptic damage. The exciting implication here is that by targeting this specific pathway, we might be able to develop new therapies to preserve neuronal function and improve cognitive outcomes for CKD patients. Of course, these are initial findings from an animal model, and the next crucial step will be to translate and validate these insights in human populations. Up next, our 10th paper out of 10, MIR 375 in CFS-derived EVs promotes HCC progression via regulating RASA1 and may potentially serve as a biomarker for HCC by Wei Qin and Songlin Peng from Department of General Surgery, the 7th affiliated hospital of Sun Yat-sen University, Shenzhen, China. This paper was selected because it significantly advances our understanding of hepatocellular carcinoma progression by elucidating a novel mechanism involving CFS-derived extracellular vesicles and MIR 375, offering both potential therapeutic targets and a new biomarker for. Let's break down its key contributions. Hepatocellular carcinoma, or HCC, remains a significant challenge in oncology. We know that the tumor microenvironment, particularly cells called cancer-associated fibroblasts or CAFs, plays a crucial role in how tumors grow and spread. This study aimed to uncover how these CAFs communicate with other cells to drive HCC progression, specifically looking at tiny packages called extracellular vesicles and a molecule called Mir-375. Our researchers designed a study to investigate this complex interplay. They used various cell models, including cancer-associated fibroblasts and endothelial cells, to track the movement of extracellular vesicles. They specifically focused on a microRNA called Mir-375 and its impact on a protein called RASA1. The team also analyzed plasma samples from HCC patients to see if these findings translated to a clinical setting. The study yielded several exciting results. They confirmed that CAFS-derived EVs successfully delivered Mir-375 to endothelial cells, and this directly led to the regulation of RASA1, a key protein. Crucially, this mechanism was shown to promote tumor angiogenesis. Furthermore, in HCC patients, those with more advanced disease exhibited higher levels of Mir-375 in their plasma EVs, suggesting its potential as a biomarker. This research significantly advances our understanding of HCC progression, highlighting the pivotal role of CFS-derived EVs and Mir-375 in tumor angiogenesis. The discovery that plasma EV Mir-375 levels correlate with disease severity opens up exciting avenues for developing non-invasive diagnostic and prognostic biomarkers. Looking ahead, these findings suggest that modulating Mir-375 or engineering CAF-derived EVs could offer innovative therapeutic strategies to combat hepatocellular carcinoma. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.